Rakovina is a computational drug development company focused on innovative cancer treatments. We have Executive Chair Jeffery Bacha joining us to walk through the story and highlight their low capital growth plans using out-licensing to de-risk drug development pathways for Rakovina. I'm Martin Gagel with Market Radius Research. It's Tuesday, September the 24th. Please remember, this is neither a recommendation nor investment advice. We are here to learn about the company. Jeffery, thank you very much for joining us. Appreciate your time. And why don't you tell us how things are going at Rakovina?
Well, thank you, Martin. It's a pleasure to be here and look forward to sharing the story with your audience. Rakovina Therapeutics is a company based in Vancouver, British Columbia that we founded in 2021. It was in part a spin out of a private U.S. company that we put some assets together with the CPC and basically launched as a public company on day one. So we're traded on the Toronto Venture Exchange under the symbol RKV. And just people often ask where you got the name of the company, and Rakovina actually means cancer in Czech and Slovak and some other Eastern European languages, which is my background. So that's where it came from. And we've been involved in myself and our team in drug discovery and drug development for many years, and in cancer in particular, because we know that it's something that affects all of our lives, all of our families, and we really hope that we're able to do something different here. And the differentiator for Rakovina Therapeutics is a collaboration that we recently established to bring artificial intelligence into the mix, into the drug discovery side of the equation. And we believe that this is very important and a way to reduce the risk, cost, and time of getting to the right drug candidate that we could advance to clinical trials with partners and hopefully be game-changing for patients. This is about speed, and it's about leveling the playing field with others who are trying to do the same thing. Speed is important because for every month that the treatment of a patient is delayed, that equates to about a 10% increase in the likelihood of that patient dying from cancer. And in particular, in the cases of recurrent disease, or when patients have their tumor has recurred following the available therapies, bringing new treatments to market is increasingly important. And this is really our focus at Rakovina Therapeutics. So when we established the company in 2021, we were a very traditional drug discovery, lead optimization, find a molecule, optimize it against a target, move it toward human clinical trials, the same thing that we've all been doing for literally decades. And we have a lead molecule that we developed through that process. Roughly 4 years and $4 million went into that effort. And we're fortunate to have a lead molecule that we're formulating toward moving toward clinical trials, and we're likely to find a partner for that sometime soon. But what happened earlier this year was an opportunity to leverage not only the relationships and the laboratories that we have in the Mads de Gaard laboratories at the University of British Columbia, but partner with another professor called Art Cherkasov, who is arguably one of the top AI-driven medicinal chemists in the world. And being able to leverage that platform and be game-changing in terms of how we discover and optimize new drug candidates to move more quickly and more efficiently and more cost-effectively toward human clinical trials is really, we believe, game-changing. Rakovina Therapeutics, we don't consider ourselves an AI company. The star of the show, what is going to deliver value for shareholders and make differences for patients, are the drug candidates that come out of the AI. The AI is simply a tool to get there better, faster, and more efficiently. I think many of us have seen this kind of a chart here where it's a really, really long time to develop a new drug, 14, 15, 16 years in many cases from the time that you have a target identified to the time that something actually comes to market and can impact patients. And potentially billions of dollars. There are stories of $4 to $6 billion to develop a new therapy. And a lot of that time and a lot of that money And most of the risk is frankly in the early stages when you're trying to find the right drug candidate to bring into human clinical trials. If you have the right drug candidate as you start your treating patients in your phase 1, 2, and eventually a pivotal trial, that's kind of a blocking and tackling regulatory process. If you have the right molecule, it is fairly straightforward and relatively low risk. The challenge is that if you don't have the right molecule, you're going to find that out eventually. The project's going to fail. It eventually will fail in clinical trials after spending millions and millions of dollars and a lot of time to find that out. So getting to the right molecule is critical. And so traditionally, we have spent 4 to 6 years working on that sort of process, exactly the way that Rakovina Therapeutics initially started. And the deck, I believe, has been stacked against smaller companies because with limited resources, we're not able to look at very many potential drug candidates. We'll look at several thousand instead of several million if you were at a larger company. And if you think about this, finding that right drug candidate is kind of like looking for a needle in a haystack. And if I'm only looking at a corner of the haystack, then the chance of me finding the right needle is limited. A larger company is going to look at a larger portion of the haystack, probably have a higher likelihood of success. Bringing the AI to the table lets us look at the entire haystack, literally screen billions of compounds against the target we're interested in to optimize them initially in silico with the computer, and then be able to have the AI suggest to us the best potential, best-in-class drug candidates that we can move forward with partners or into the clinic ourselves. And this doesn't take years and millions of dollars. It takes weeks. And that is game-changing, in my opinion, to how we're approaching this. So we've been very fortunate to be able to collaborate with Professor Cherkasov and the novel deep docking platform at the University of British Columbia. We also recently expanded our AI efforts to include a partnership with a company called Variational AI, which is also here in Vancouver. And Professor Cherkasov is actually an advisor there. And they have a certain expertise which dovetails into some of the targets that we're interested in. So it was a natural fit to expand that relationship. So the modeling happens in a very rapid way to identify those drug candidates. And then we bring them in-house, validate them in our labs, and we'll be looking at partnering. So our focus is something called DNA damage response and or DDR. And these are pathways in our cells and mechanisms in our cells that are designed to protect us. They detect and repair damage to our DNA on an ongoing, regular, seamless basis. If it's a sunny day outside, we just got through summer, and I'm sure we were all enjoying a lot of nice sunny days outside, whether we're at a patio or what have you. You're getting hit with UV light, but you're not coming back to your office or going home after the day with cancer. There was damage happening from that UV light, but these systems were detecting it and fixing it without us ever knowing. It happens tens of thousands of times a day. What we've also learned is that in about 3 out of every 4 cancers, there's a defect in one of those systems that allows a mutation to sneak through. And if that mutation is associated with cell division or cell growth, eventually a tumor is going to form, and that person is going to become a patient who's diagnosed with cancer. So this is learning that's happened over the last 30 plus years, which creates a really interesting opportunity for using artificial intelligence, because all of that information, all of that learning about these systems from patents, from publications, from FDA minutes can be used to train the system. And what we're asking the system to do is to find us, based on all of that information, novel, best-in-class therapeutic candidates that are not in anybody else's patents, which we can then own. We've really just frankly scratched the surface of this opportunity to impact patients. There is a first-generation class of drugs in the DNA damage response space called PARP inhibitors. They're sold by GlaxoSmithKline, AstraZeneca, and Pfizer, some of the biggest companies in the world. And interestingly, none of those companies invented those therapies that are in the market. They got them by licensing them from another company. or by acquiring a smaller company. And that's certainly the pathway that we believe that we will be on as well. Our first target in the space is based on the success of the first generation drugs, which are called PARP inhibitors, PARP. They have become game changing for certain types of breast, prostate, and ovarian cancer. They're very important drugs. Again, results of decades of research before they eventually reached the market, starting at just a few years ago. And like any new technology, it's amazing when it first hits the market. It does amazing things for in this case patients, but we learn from that. And like any first generation technology, you start to learn things and ways that you might be able to do better. So in this cases, there are some side effects that are associated with these molecules, these treatments. And they also are not very good at chasing a cancer that has spread to the brain. And that's important here because about half of the breast cancer patients who are being treated with these first generation PARP inhibitors will experience the tumor spreading to their brain during the course of their treatment. And right now, that's a tough thing for us to deal with. And usually that's what is going to result in the patient succumbing to the disease. So being able to reduce the side effects and to be able to cross the blood-brain barrier is something that's very important here. And AstraZeneca is clearly the leader in the field here, and they have a second-generation drug that is likely to be approved by FDA in the next 18 months, and that is focused on reducing the side effects. So they've focused specifically on PARP1 rather than having a molecule that hits a number of PARP enzymes, and their data is quite wonderful. And there are a number of other companies that are working towards something similar. AstraZeneca also has what I'll call second generation plus in their portfolio, which is a PARP1 specific, so reduced side effects, that has the ability to cross the blood-brain barrier. And that looks to be quite game changing. And that's what we are looking for as well. We are looking for PARP1 specific CNS penetrant, so cross the blood-brain barrier, a molecule that is as good or better than what they are developing. And we know that there are an audience of potential partners in the other companies that are in the field that are looking for the same thing. And our goal is to use the AI to get there in a very efficient and quick manner and be able to potentially partner and license to those other players in the field. So our timeline looks something like this. We established the initial collaboration with the deep docking platform in the spring, began training the AI, which is going to be an ongoing process. We announced in our quarterly filing recently that by the end of June, we had screened roughly 1.5 billion compounds against the target that we're looking for, against that PARP target. That'll eventually move up to about 7 billion. It's not going to take months to do that. The system actually learns and moves faster as we go. And so we expect to have the initial output, the initial core sample from the mining exercise against the PARP target in the early fall. So we expect to be announcing that we've received that in the next few weeks. From that point, we will begin bringing those compounds in-house. We'll be synthesizing them with our chemistry partner, who's a company here in Vancouver called Farm Inventor. We announced recently that we had expanded the relationship with them to be able to move more quickly to do this. And as they synthesize those, we'll be bringing them in-house and testing them and validating them in our in-house laboratories at the University of British Columbia, which has been a wonderful relationship and has really allowed us to have a platform to make all of this happen. As we're validating those, we anticipate presenting the initial data here from this effort at the Society for Neuro-Oncology annual meeting, which is in November. This will be the first time we're showcasing the data that's coming out of this. This is also a great opportunity for us to continue the conversations with the potential partners that we will look to help us move these into clinical trials. And our goal is to have a partnership in place by about the middle of next year. The partnerships we're talking about, the size of these investments made by big pharma into the DNA damage response space are significant. There are multiple examples of preclinical deals, license agreements before human clinical trials ever start in this field that are in the billion-dollar range with very significant upfront payments. The most recent example is from about a year ago. The Merck KGaA, the German Merck, licensed one of those PARP-1 specific programs from a Chinese company for $1.5 billion with $160 million up front. If we're successful in achieving a partnership like this, we would take that initial capital and use that to build out our infrastructure to be able to go after multiple targets simultaneously and really make a big difference for patients. So the team around us, myself, my background's biophysics. I've been building companies in the oncology and other biotech fields for a long time. I'm blessed to have relationships with some wonderful people in the laboratory. Mads de Gaard, who's a professor at the University of British Columbia. Mads has been a go-to person for projects that I've been involved with for many years in terms of how does this drug interact in the cell? What does it do with DNA? His expertise is fantastic. And when we were putting together Rakovina Therapeutics, Mads was helping with some due diligence and got excited and said, "Hey, can we do this in my lab? Can we, can I be part of the company?" And so we worked it out with UBC that, he's a professor at the university, but he's also president and chief scientific officer of Rakovina Therapeutics and has a dedicated team in his lab that is responsible for the work that we are doing. The partnership with Professor Cherkasov, who I mentioned, is really born out of a friendship between Art and Mads. Their offices are next to each other. And, over time, they started brainstorming about the ideal nature of pointing a deep docking novel AI platform to DNA damage response targets because of all of the information that we have to train the AI. John Langlands, our chief operating officer, has played that role in every company I've been involved with in the last 25 years. So this is a team that's worked together in the past very successfully, and we know each other all very well. Al De La Crazia is our vice chairman, independent director. One of the key things that is important is having a good team around you on the financial side. And when we were putting the company together, we looked at a number of CPCs to help fund this and put everything together. And Al led a team that had been successful in the mining field, not in biotech, but their track record of sticking behind things that they got excited about, being major participants in the market and supporting the company. This was the same group behind a company called GT Gold, and that CPC group never sold shares all along the way until that company was acquired. And there's some wonderful people in that group who have been very supportive of Rakovina Therapeutics. And that's what you need around you when you're building a company. And we're very grateful for it. Advisors, Dennis Brown is a collaborator and a partner of mine for many years. He and I are involved in a private company called Edison Oncology in the US, which is the major shareholder of Rakovina Therapeutics. Dennis has a track record of many exits successfully over the years. Petra Hamerlich joined us just a few months ago on our scientific advisory board. She, prior to going back to academia, was the head of the DNA damage response programs at AstraZeneca. Was responsible for bringing forward what is currently the market leader in the DNA damage response field, the PARP inhibitor olaparib. She's also responsible for their next generation drug, the one that's going to be approved here by the FDA very likely in the next 18 months or so. Their phase 2 study that AstraZeneca presented was called the Petra trial. It was named after her because it was her project. So when she went back to academia, we were able to eventually bring her on board onto our scientific advisory board. And frankly, having somebody who's got the institutional knowledge from one of the leaders in the field is very, very important and a blessing to have. Len Post, also involved in the original first generation drugs, the product that in the DNA damage response field that Pfizer sells, palazoparib. That's Len's drug. He had it at Lead Therapeutics. He took it to BioMarin. It went to Medivation, and eventually Pfizer bought Medivation for $14 billion and commercialized that product. So around us, we have advisors that are just fantastic, that are helping us move everything forward. So that's really the story. Great people around us. A very large market in terms of DNA damage response opportunities where we've really just scratched the surface. These are programs over the years that Rakovina Therapeutics, and other companies working in this field hope to impact 3 out of every 4 cancers. And the way that we are doing that is to leverage AI to move as quickly and as efficiently as possible to hopefully be ahead of the competition, to identify those best-in-class therapies that can be validated in our world-class laboratories at the University of British Columbia, and then partnered with major companies to bring forward. So, Martin, thank you very much. That's really the story, and look forward to any questions.
Excellent. Thanks, Jeffrey. Yeah, a lot of questions come to mind here. Just to clarify, you have your initial lead compound, which you developed through the traditional test tube manner, call it, sorry, that's a crude way of saying it. And now you have this other one starting down the pipeline, which is an in silico in the computer being developed. Is that correct? Those are the two sort of current.
Right. So the pipeline has evolved like as follows. So the lead program was born out of the original lead program, let's call it, was born out of the more traditional drug discovery and development methods from our 3000 series. And that is a program that hits not only the PARP target, but also a second target that is correlated with resistance. So it's a dual targeting class of molecules. And what we've shown is that when resistance to those first generation products in the market from Pfizer, AstraZeneca, and GSK, when those drugs work for a while and then almost without fail, over time, the tumor becomes resistant to that and will tend to come back. So that program was designed to address that. And like I said, we're very lucky to have a lead molecule there that we are in the process of formulating toward the clinic. What we've done since then with the AI is initially to say, let's look at what's happening in the field, and the desire to have in those first generation compounds, first generation products, let's reduce side effects and be able to cross the blood-brain barrier. So we're very focused there now with what we call the 2000 series, and that's the AI driven. Beyond that, our partnership with variational AI will allow us to begin expanding our research into kinase targets. That's their expertise, and it made sense since we have a lot of overlapping relationships to leverage that and bring that into the fold as well, because 3 of the key targets in the DNA damage response field are kinases, and that so it was a perfect fit.
The computational drug developed, that is not superseding the original drug developed. The initial one still has sort of a pathway of its own.
Absolutely. The initial program is valuable. We believe it's important for patients, and we're pleased with the success we've had there. It's just that allowed us to establish the laboratory infrastructure, that expertise in PARP in particular, that we're leveraging now with the AI in the 2000 series program.
Okay, so let's just stick with that drug. Your overall model, at least initially, due to the capital constraints of trying to run your own trials, your strategy for the initial drug is to out-license it to a partner. Is it at the stage where you're in negotiations for that? Does it need to be further developed before you can do that out-licensing?
So, in terms of the AI programs that we're looking at, our goal is to partner.
Okay.
And frankly, the idea, as I've said, the attractiveness of the deals that have been done preclinically in this field is it's striking.
Yeah.
And so to be able to say we can accelerate the development with a larger company, and unlock value in terms of a large upfront fee that we can use to build the company with non-dilutive capital is something that is, I think, it's a very attractive business model and proposition. So the concept of being able to partner early and leverage that capital that comes from the partnership to build out the company and go after the next target. And then eventually maybe bring something into the clinic ourselves, but for now, the opportunity to partner early and hand something off or collaborate and work together with a player that has the infrastructure to run large clinical trials, do the manufacturing necessary for those trials and for the market, is very attractive to us.
All right. So the initial drug, is that in the out licensing negotiation phase right now, or are you still doing additional work on
So
that?
the 3283, the dual function inhibitor that came out of the initial work,
Yeah.
we're optimizing, excuse me, not optimizing, we're formulating that to be able to be advanced to clinical studies. And we are, of course, talking to potential partners around that. And we're hopeful that we'll be able to collaborate with somebody to move that into the clinic. With the AI-driven programs, we announced the initial collaboration with Professor Cherkasov and the deep docking platform the week before the American Association for Cancer Research annual meeting. And the reason that we did that was that we know that every big pharma company out there has a robust DNA damage response research and development program in-house. And to be able to go to that meeting with that collaboration in hand and say, we are looking at changing the game on how we do drug discovery. We're going to leverage what is one of the most well-validated and successful AI platforms in the world, and we're going to have exclusivity toward our targets. We want to talk to you about what you're looking for. And those 8 companies that are chasing AstraZeneca in the PARP space, it's very clear what they're looking for, and that's what we are hoping to deliver. But being able to go with that announcement in hand, so we could talk openly about our approach and what we were doing, and then going to the bio meeting in June and being able to follow up with that. Those conversations, and then of course, hopefully having some exciting data to share at the Society for Neuro-Oncology meeting in November. Those are conversations that are evolving over time, and keep in mind, this is something about this platform. The validation here, there are multiple drugs in clinical trials that were driven by the deep docking platform. The largest deal that's ever been done by a Canadian university with a pharmaceutical company is something that was driven by the deep docking platform. So the validation here, including what was done during COVID to screen literally 1 billion plus compounds in 3 weeks against the COVID virus, which led to multiple programs that eventually impacted the virus. The validation of this platform is unquestioned, and we're very fortunate to have the relationship that gives us exclusive use of the platform toward the targets that we care about in the DNA damage response field.
Once you get the sort of the computational output, okay, this molecule, that, then the next step is you go to one of your partners and they actually produce some of the molecules physically, where then you do some test tube testing on it, so to speak.
That's correct. So we have a wonderful chemistry partner here in Vancouver called Farm Inventor that we've worked with for years. We recently expanded our relationship with them to enable us to go faster, because we knew that this was going to be happening with the AI. So what the output of the AI is intended to be is a short list, a curated short list of recommended molecular structures. Think of it as almost like a mining exercise. So we've got the real estate that we're interested in, which is that PARP target, and we know what we're looking for, which is that PARP1 specific DNA or brain penetrating profile. And so that first mining exercise in the core sample that's delivered, we're not looking for uranium or gold in that core sample, but it's that short list of curated, ranked structures that are recommended to move forward. And so we will work with Farm Inventor to synthesize those, and that'll happen over a few months. But as each one is synthesized, that will come in-house, and we've got the infrastructure, as you said, in the test tubes, but also in the cell-based assays, in the toxicology assays, pharmacology, the pharmacokinetics, metabolism, and animal models, all in-house, that we will use to validate those. And those are the data, being able to show that we can treat that cancer, that the drug is well tolerated, that it can cross the blood-brain barrier, the profile that we're looking for, and deliver those data of a validated new molecule, new class of compounds that we have patented to a pharma partner is what we're after.
Are you going to be doing all the preclinical work yourself, or would that be out licensed before you get the all that preclinical work done?
The majority of, well, majority if not all of the preclinical work for the 2000 series will be done in-house. We have the infrastructure established from the work that we did originally with the 3000 series. That's been established, and literally we have access to hundreds of millions of dollars worth of infrastructure through our collaboration with UBC and the work that we're doing in Professor Dugard's lab that lets that happen. And so it keeps our timeline short because we're not waiting for a CRO to perform work for us and give us data. It happens in real time. It's down the hall. And it also keeps the cost relatively low because we're not paying CRO overheads in order to make that happen. So it's let us do things very efficiently and very quickly. And that will continue to play within what we're doing with the AI.
But this isn't the lab that you spent millions of dollars building yourself. This is a UBC lab that you're sort of getting the benefits of the scale of a CRO, but a world-class university, and then getting the work out of them.
We're getting the benefits of a large pharmaceutical company through the university. It's not just a CRO because CROs tend to be very specific toward what they do. The university has sort of a soup to nuts big pharma type of infrastructure that we are able to benefit from through our collaboration, and it's a wonderful way to move things forward.
All right, and then in November you're going to present a poster at the conference with your initial digital discoveries and just sort of highlighting maybe their characteristics or the broad scope of them, and to sort of show off what you came up with?
We'll show off the best of what we have at the time. Exactly what data we'll have in hand remains to be seen. But the idea here is to use that Society for Neuro-Oncology meeting. Yes, it is a poster presentation, but that's nice because as opposed to standing on the podium, you have several hours of time with your data sitting there that you can interact with not only clinicians, but those potential pharma partners, asking questions, showing them the data, walking them through it. So it's a very good opportunity for us to continue that ball moving forward. And we plan to present the most advanced data that we have at the time at that meeting.
One of the benefits of the strategy you're doing of out licensing it relatively early is that it's a lot less expensive for running the whole drug development. Can you talk a little bit about what sort of the expected costs would be from sort of start to out licensing and doing the initial preclinical stuff at UBC with your partners there?
Right.
What would one expect that to cost?
Well, the historical setting, with the 3000 series, as I said, is, 4 years and $4.5 million to get to a molecule that we're now ready to formulate. And then going from there, you're looking at, an investment in formulation, an investment in scale up, which can be anywhere from the hundreds of thousands to the millions, depending on the program. And then getting into clinical trials, the initial clinical studies, you're looking at $2 to $5 million for a phase 1-2 study, possibly a little bit more. And then if you're getting into a pivotal phase 3 study, that can be tens to hundreds of millions of dollars. And that's a daunting task from a young company's perspective. And being able to have the opportunity, particularly in this field where big pharma has been actively investing in and partnering in a preclinical setting, allows us to unlock value of the drugs or the assets that we are developing without having to make that big investment in clinical trials, for example.
So it took the initial one that you said about $4 million in 4 years. What would be using your new model or your new approach, about how long? It sounds like you'll have good data out later, early next year or so. So it turns it into what, 1 year and $1 million
The model we've been working with is the That initial mining exercise to get to that core sample is 12 to 16 weeks, so 3 to 4 months. And then from there, 6 to 9 months of moving forward toward having the data in hand that can be partnerable. So it's really become a 9 to 12 month exercise rather than a 4 to 5 year exercise to get to that molecule. And the cost overall is a fraction.
Curious about your training data set for your AI model. Are they all novel synthetic compounds?
So the training data set is generally public domain information. So information in patents that have been filed in the last 20 plus years of DNA damage response research, publications, particularly around molecular structures that are more likely to cross the blood-brain barrier. Information from FDA, information from conference presentations and publications. All of that information is used to train the AI. And the question that you ask of the system is, find me something that looks like this. Here's the profile I'm looking for. In this case, it's the PARP1 specific blood-brain barrier cross molecular structure. Recommend to me structures that you think will do that, that are not in anybody else's patents, because part of the training is the patent, so we know the structures that other people own, because we don't, we want to have something that we own and we can patent that is novel.
And I guess it's just like using Chat GPT. You need to know what question you're asking it to solve. You need to give it the right prompt. And I'm sure it's not a 1 or 2 sentence thing. You really need to, you need smart. I couldn't discover something because I wouldn't know what kind. I couldn't just say cure this. You have to say, these are the angles we're trying to do. This is how we're trying to approach it.
And that's quite an important point to make, because the AI has it's artificial intelligence. It's got a mind of its own, if you will, but there's still an operator who's driving that system, and having a very skilled medicinal chemist at the helm in Art Cherkasov is one of the drivers of success. And that is why the platform has been so successful in the past. And there are multiple products in clinical trials, big deals that have been done from this platform, as well as the impact in COVID. And that has as much to do with the robustness of the system, which we believe is head and shoulders above anything else that's out there, as it does with the architect of the system and the individual who's overseeing it. So it's a combination of human ingenuity and knowing the field and what you're looking for from a medicinal chemistry standpoint, and letting the system do what it can do.
Another question here. Is the in-house validation of targets done in collaboration with the academic labs at UBC?
Well, certainly. We do a lot of collaborating with the UBC. So when we're looking at things like the PARP target, this is already a validated target. There are 3 drugs on the market that are PARP inhibitors that generate about $3.5 billion a year in sales currently. So that's a well-validated target. And like I said before, these first generation products that are out there have been frankly game changing for patients. But like any newer technology, it's amazing when you first see it, and then you say, what can I do better? If you think about having in your hand an iPhone 6 and an iPhone 14 or whatever number we're on now, they look pretty similar in your hands, but they do very different things. And part of that is driven by the evolution of the engineering and the ability to do things on a chip that you couldn't have done in the iPhone 6 age. And biology behaves the same way. We learn things about how to manipulate cells, what happens in a cell with a drug, and we can dig deeper into that understanding today than we could even 5 years ago. So that allows us to say, well, these first generation drugs were very important for patients, but let's do better, and let's leverage our knowledge that we have across the field, and hopefully deliver more important therapies for patients that solve important problems.
Does a computational model take into account safety and tolerability, or that's one of the constraints? Okay, it needs to do this, but also take those
Well,
parameters in?
certainly, we all know that some certain toxicities happen with drugs, and so the molecular structures that are associated with those, binding to certain targets that are associated with those. So you want to have a drug that hits your target, but not a bunch of others is the ideal. And so all of that can be built into the algorithm and has been established in the deep docking algorithm. Beyond that, there are certain features of chemistry that lend themselves to stability in the metabolic environment that have the drug be able to survive long enough in the body to get to the tumor and do its job. The pharmacokinetics of how much is absorbed, how quickly. I mean, all of those things are information that has been built over generations and can be used and has been used with the deep docking system. So we're not just looking at does this bind to the target, but the structures that are recommended based on safety profile, metabolic profile, pharmacokinetic profile, all of those things that are going to make it likely that the short list that we're seeing are drug candidates, not just interesting things in the lab.
Are you looking at PARP1 specifically, or have other or do you have other targets in the pipeline?
Well, initially it's been PARP, but through our collaboration with Variational AI, their expertise, as I mentioned, is kinases, and their platform is built on kinase drug discovery. And there are three key targets within the DNA damage response field, ATM, ATR, and DNA PK, that happen to be kinases. Kinases are switches in cells. They turn things on and off. And in the DNA damage response field, three of the important targets are kinases. The Variational AI platform is very much trained on kinases, and so the relationship with them is to have some exclusivity specifically against those targets. So as we go forward, the next things beyond PARP for us will be out of that list.
And I want to circle back to your partnerships you have with the different, you have numerous partnerships. How are you accessing their knowledge and their technology? Are you paying them sort of by the hour, or are they sort of getting they get some sort of a license or royalty on what they contribute to? How do they get sort of their end of the deal?
It generally varies across the board. So we have a relationship with the University of British Columbia. We have a contract that says every 6 months there's a payment made, which funds the people, the laboratory infrastructure, and all of this. Or on the other hand, for example, with Farm Inventor, we have full-time equivalent employees at their chemistry facility that are dedicated to our programs. Building out a medicinal chemistry lab is a big infrastructure to do, and they have a wonderful infrastructure that we're able to work with. So it varies, but we put these things, these partnerships together in a way to hopefully accelerate our research in a way that gives us always control and ownership of the drug candidates that come out of them.
So if I have it right in terms of milestones coming up, you're going to, in sometime relatively soon, you're going to be announcing that you've generated a short list, and then that you'll be going to this conference in November and presenting your data. Can you walk us through what milestones investors should be looking for over the coming months and quarters, and what to be keeping our eyes open for?
Well, sure. So, like we have on the slide here, this is the deep docking program toward that PARP1 specific brain penetrating best in class molecule that we're going to look to partner. So, as you mentioned, we expect to have the initial core sample from the PARP mining exercise in the early fall here. So we anticipate announcing something in the next few weeks. The Society for Neuro-Oncology meeting is in the third week of November, so we will be having an announcement there. We are also presenting some data around the 3000 series, the original program, at a couple of conferences in the fall. So we'll be talking about that data as well. And then, of course, with the variational AI program, that's going to move very rapidly. So when we choose one of the targets that we have exclusive access to, they'll deliver their shortlist in a relatively short amount of time. And then we'll, over the next 12 to 18 months from there, be doing the same exact thing we're doing here in terms of chemistry and validation in the laboratory. So there will be a number of milestones, both in the KT2000, the Part 1 specific program that we're talking about on the screen here, as well as through other programs that we'll be reporting. But specifically on the deep docking core sample announcement that we've received that from the initial mining exercise is in the next very little while. The key data presentation at the Society for Neuro-Oncology meeting in November, and then eventually, hopefully by about the middle of next year, announcing that we have formed the partnership that we're looking for to take that program forward.
You're obviously going on a lower cost model of drug development. You've recently raised a little bit of some money in the marketplace. Can you talk about your runway, how far that gets you, and what should investors be expecting on that front?
Well, ideally, the runway that we have gets us to the partnership that we're looking for. Obviously, we want to make sure that we have a strong balance sheet as we go into those discussions. And so, opportunities to raise additional capital between now and then, we'd certainly be enthusiastic about.
But if you keep your belt tight, you can get to sort of the end game as you're structured currently. But obviously, having a little more buffer would be nice.
Right. It's having having a stronger balance sheet is helps to keep the conversations with the potential partners as honest as possible,
Yes.
because your back's not against the wall. So we're certainly enthusiastic about the support we've had from our shareholders, and have been introducing the company to new parties on a very regular basis, increasing the number of people who are aware of the story. And so we're hopeful that there'll be opportunities to raise additional capital as we go forward.
I think we've covered, we've done a pretty good dive into Rakovina. Is there anything we should or you want to bring up before we wrap things up here?
No, I think just reiterating, the big picture here. This is a massive opportunity in the DNA damage response field to impact patients. We've only just scratched the surface with the first generation of programs that are on the market, and we believe that we can do better in that setting, in the PARP setting. But there are other targets that are important here that we can go after, and we believe that by partnering with the leading AI firms that we have with the deep docking platform and variational AI, allows us to potentially get to that best-in-class lead molecule ahead of our competition. We can validate that in our laboratories and then have opportunities to partner with that competition in a very attractive manner. That's our goal, and we look forward to sharing progress that we made toward it.
Jeffrey, thanks a lot for coming on the show, explaining what Rakovina is up to, and it's exciting to watch. Lots of milestones hopefully coming up here, hopefully create lots of value for you and the shareholders. So thank you very much.
Good. Thanks for having me, Arden, and look forward to staying in touch.
Cheers. Talk to you soon.